In our last topic, we discussed signaling systems in the body. Specifically, we talked about the endocrine system. The endocrine system, of course, is going to dump hormones into the blood, and the hormones serve as signaling molecules. Because the hormones are dumped into the blood, they can get everywhere in the body. So they diffuse out of the blood into the interstitial fluid. They have the potential to make contact with any cell anywhere. Cells are only going to respond if they have the appropriate receptor. We don't need a whole lot of hormone to make this work because, of course, if a hormone binds to a receptor, the response is amplified within the cell. Now, there are a few drawbacks to the endocrine system. Despite how efficient it is, it's a slow-acting system in some cases. It takes a little while for hormone levels to build up, and it takes a while for the hormone to be removed. So once the endocrine systems start to bring about responses within the body, the only way we can stop those responses is to get rid of that hormone, to pull it out of circulation, and that can take a little bit of time. Now there is, of course, another signaling system, and that's the nervous system. The nervous system is a very energetic system. So we lose some efficiency in the sense that we need to use a lot more ATP and a lot more resources for this particular signaling system. But it's very quick and it's very precise. So nerve cells can conduct electrical signals known as action potentials. They do this very rapidly, and nerve cells can synapse or connect with very specific cells, cells that are referred to as effectors. They bring about an effect, and those would be glands and muscle cells and other cells, as we'll see. This is a big topic. We've got lots of stuff to cover, so I've divided it up into two parts. In part A, we'll look at the anatomy and also the development of the nervous system. In part B, we'll look at the physiology. Specifically, we'll look at how action potentials are generated, how they travel along neurons, and how that signal is relayed across the synapse to other cells. Let's start at the beginning, quite literally, by looking at the development of the nervous system. You started out as a single cell. So your father's sperm and your mother's egg came together and created a diploid zygote that then underwent many rounds of mitotic division. These early divisions are known as cleavage divisions. So the zygote is going to divide or cleave into two cells. Then those cells will divide into 4 cells. Those cells divide again and we end up with 8 cells. And you can see in this diagram at the top, we have this solid ball of 8 cells, something known as a morula. Don't worry about that, but in case you come across that term, that's what it's referring to, this solid ball of cells. Those cells divide again, and we have a ball of cells that contains 16 small cells. Notice that the cells are getting smaller with each division. After that 16 cell stage, something a little different starts to happen. The cells continue to divide, and we continue to call those divisions cleavage divisions, but the ball becomes hollow. So the cells move towards the outside, and we end up with this hollow ball of several hundred cells known as a blastula. What happens next is a process known as gastrulation. It's called that because it creates something called the gastrula. Basically, one side of this hollow ball is going to be pushed in. When that happens, we have the creation of a new layer of cells. So we have that outer layer of cells, and that's now known as the ectoderm, ecto meaning outside, but now we have this inner layer of cells that's known as the endoderm, endo meaning inside. And we have an opening. That opening is known as the blastopore. The cavity that was within that hollow ball is known as a blastocele. Remember, cele, C-O-E-L, means cavity. And that little bit that got pushed in, or invaginated, got pushed into that blastocele. Now we're going to have another layer of cells that's going to form within that blastocele that we'll talk about momentarily, that's known as the mesoderm. Now, one thing to note, this is what happens in all animals. This is what happens in worms and starfish and insects and mammals like ourselves. But in vertebrates, so mammals and fish and birds and etc. Things are a bit more complicated because the ball is kind of squished and it sits on top of a yolk sac. And that's what you're seeing at the bottom there. What you're seeing at the bottom is how this process occurs within a reptile or a bird. Basically, within the egg that they lay, there is yolk, and on top of that yolk, there's this little disc of cells. But it starts in the same way. We start with a zygote, we have cleavage, we get this blastula, but it's kind of a flattened disc. So imagine taking the blastula and squashing it against this yolk. Same process happens, but now we have this groove that forms along that disc. This is where cells are moving inwards, a process known as invagination. Those cells are moving inwards and they're giving rise to our three layers, our three primary layers. And those primary layers are known as germ layers. Germ, unfortunately, means a lot of different things in biology. But we have our three primary germ layers, the endoderm, the ectoderm, and then the mesoderm develops a bit later in between those two layers. This process is further complicated by the fact that many vertebrates produce extra-embryonic membranes during development. These are membranes that surround the embryo and are produced by the embryo to help protect and maintain the embryo. At the top here, what you're seeing is a typical egg, the kind of egg that might be laid by a chicken or a reptile. You can see that we have these structures and membranes that are produced within the egg by the embryo as it develops. So we have something called the amniotic sac that contains amnion. This is a fluid that supports the embryo. We have a yolk sac, and the yolk sac is going to contain nourishment that was provided by the mother. And you can see it's connected to the embryo by blood vessels. We have something called an atlantos, which is for waste. So of course, if you're trapped in an egg, you can't get rid of the waste. Instead, you have to store it until you hatch, and it's stored within that sac. A few other interesting structures. We have something known as the chorion, which is this protective membrane around the outside of all those other structures. We have a space which is used for breathing. So there's a little cavity here at the end of the egg, which you've probably noticed if you've peeled a hard-boiled egg. And that's where gas exchange is going to occur through the shell. Now, why am I telling you all this? Well, first of all, I'm going off on a tangent. I find this stuff fascinating. I'm not going to ask you about this stuff, but it's stuff that we will come back to when we get to development next term. But anyway, the other thing that's really fascinating is that mammals are also considered amniotes. So amniotes, again, are reptiles, birds, but mammals as well. And you might be wondering, well, why would a mammal be considered an egg-laying vertebrate? We don't lay eggs, unless, of course, we're talking about platypuses and echidnas. Well, within the mammal, the egg is retained. So basically, we have the exact same structures that are produced by the embryo, but they're produced while the embryo is inside the mother. So we have a yolk sac, we have an atlantos. Together, these make up part of the umbilical cord. And then there's this new structure known as the placenta, which is built by the coordinated efforts of the embryo and the mother. I wanted to take a bit of time to explain where these extra-embryonic membranes come from so that the next series of diagrams make sense to you. What you're seeing here is a very early human embryo. This is around the stage where the embryo would implant into the lining of the uterus. In the first diagram, we've got this disc of cells here, and this is the actual embryo. Above that, we've got the amniotic membrane that's going to form the amniotic sac that will eventually completely surround the embryo. Below that, we've got another cavity that contains yolk that was contributed by the mother. Now, in the second diagram, you're seeing the actual process of gastrulation. Gastrulation begins with this invagination. Invagination refers to the creation of a cavity, the creation of a sac, basically a blind sac, a sac that doesn't go anywhere. We've got cells being pushed in, and the cells are going to develop new identities, basically, new fates as they migrate. So the cells on the outside are going to form the ectoderm. The cells that move inwards and come into contact with that yolk cavity are going to form endoderm. And then a little later on, we're going to have cells in between those two layers that form the middle layer, the mesoderm. Meso means middle. With gastrulation complete, we now have our three primary germ layers. The ectoderm is going to give rise to the skin, but it's also going to give rise to the nervous system, as we'll see in a moment. The endoderm is going to give rise to the endothelium of the digestive system and also the lining of any ducts and glands that connect to the digestive system. It's also going to give rise to epithelium within the respiratory tract. The mesoderm is going to develop into muscle and connective tissue like cartilage and bone. You can see that there's a structure within the mesoderm known as the notochord. This is a rod of connective tissue that runs the length of the back of the embryo. It's going to serve as a scaffold for the development of the vertebral column. After development is complete, the only thing that's left of the notochord is the intervertebral discs, so those cushioning connective discs between the body or centra of the vertebrae. The endothelium has completely surrounded the inside of that yolk cavity, and the yolk will become surrounded by the digestive system, and it will be broken down as nourishment and disappear. Okay, looking at the second diagram, we have ectoderm at the top shown in green, and you can see it's folding to create a groove, and that groove folds over completely and forms a tube, and that's the neural tube that you can see in the last diagram on the right. That tube is going to give rise to the spinal cord and brain. Note that we have the digestive system that consists of an outer layer of mesoderm and an inner layer of endoderm. So that would be this tube here. The digestive system is just a simple tube. It expands in some areas to form pockets like the stomach, for instance. But it's this tube with mesoderm on the outside and then this epithelial layer that's derived from endoderm on the inside. Notice that we have a coelom, a body cavity that's developed within the mesoderm. The mesoderm is shown in purple here. That would be the ventral body cavity, for instance, that consists of the thoracic and also the abdominopelvic cavities. Also notice, finally, that extra-embryonic layer, known as the amniotic membrane, now completely surrounds the whole embryo. So that has given rise to this fluid-filled sac that the embryo is floating around in. The process by which the neural tube forms is known as neurulation. What you're seeing here on the left is a very early embryo. So this embryo has completed gastrulation and it's just starting into neurulation. We're looking down from above the embryo. You can see the head towards the top and the tail or caudal region towards the bottom. Now at this stage, we have a groove that's developing along the dorsal surface or back of the embryo, and you're seeing that in cross-section to the right. The neural plate, shown in blue, is the part that's going to bend over and form the neural tube. We have a crest on either side of the groove. That's shown in green here, and this is known as the neural crest. Here you can see that the groove has deepened and the anterior part of the groove has widened considerably. The neural plate is invaginating further and we get more development of these neural folds or neural crests. The neural tube forms first in the middle. It takes longer to form towards the caudal and cranial portions of the embryo. So we get the tube first closing off in the middle, about the area where we see this cross section. Incidentally, these bumps that you see here are known as somites. They're going to develop into blocks of muscle. Now note those neural crest cells. Those are the cells that were found within that neural crest before it folded over completely. Those cells are really quite special cells. They wander around and they give rise to lots of different structures throughout the body. So they can leave that area, wander around into the skin, for instance, and they give rise to melanocytes throughout the skin. They also wander into what will become the face, and these cells give rise to the bones of the face and other structures of the face. These cells also wander off and give rise to the peripheral nervous system. So that consists of the nerves that will connect to the central nervous system. We can see at this stage that the neural groove has completely closed over to form a tube in the middle of the embryo, but it's still open at the ends. So the progression of closure will extend cranially and also caudally. And if everything goes as it should, we should get a completely enclosed tube that's expanded at the anterior end. That expanded portion is going to give rise to the brain. The other thing to note, it's not shown here, but the notochord would be developing underneath that neural tube. Remember, the notochord is going to align and manage the development of the vertebral column. The neural tube, of course, is going to give rise to the spinal cord, and it's going to end up within the vertebral canal of those vertebrae. And this is just an overview of that entire process. So again, we have the notochord, which is a rod of connective tissue. It's derived from mesoderm that runs the length of the embryo just below the surface. We have ectoderm that's going to fold in. It's going to form the neural plate, which becomes the actual neural tube. And also we have these neural crest cells at the crest of that groove that will wander off and do all sorts of wonderful things throughout the embryo. We have blocks of muscle known as somites that develop on either side of the neural tube. The neural crest cells will give rise to peripheral nerves, and notice they're also giving rise to what are called spinal ganglia. This is where the peripheral nervous system connects to the central nervous system. The central nervous system is going to come from the neural tube. Even after development is complete, there is a canal that runs through the spinal cord. It remains a tube. So we have this very small central canal that runs through the spinal cord, and it connects to cavities within the brain known as ventricles. You can see in the bottom diagram here, the first trimester embryo has basically a swollen tube that's going to develop into the brain. The brain will expand, but it will still maintain its tube-like structure. There are a number of disorders that can result if neurulation doesn't go as it should. If the cranial portion of the neural tube doesn't close completely, then that means that the cranium and the upper portion of the brain won't form. And we're left with a condition known as anencephaly. Encephala means brain, and means without. We're missing a portion of the brain. Individuals that are born with this condition generally don't live very long. Quite often they're stillborn. In many cases, they still have a functional brainstem, and that can keep them alive for days or weeks, and in very rare cases, longer than that. But because they lack the cerebrum, they also lack any form of consciousness. So this, of course, is a very serious disorder. In spina bifida, the neural tube fails to close at the caudal end. Depending on how large the area is that doesn't close over, we can have mild cases or severe cases of spina bifida. In figure A, you're seeing a normal newborn vertebra. Now, it's not completely ossified yet. There's some cartilage in there, but you can see that we have a neural arch on the top here that's going to protect the underlying spinal cord. In example B, there's a gap, and that's because there was a small gap in the neural tube where it didn't close over, and that didn't allow for the complete production of a closed neural arch. So we have this gap that only has soft tissue. And of course that can be a problem because the spinal cord's not being protected there. The other thing that's interesting in these cases is quite often there's dense growth of hair right over that spot. So that can be one of the signs that the neural tube has not closed. In C, we've got a large opening. And in fact, we've got a cavity that's formed, almost like a sac, almost kind of like a blister, that's filled with cerebrospinal fluid. And in the last case there, D, a rather serious case, the spinal cord actually projects into that sac. That needs to be operated on. Of course, the last two definitely need to be operated on. The last case there, case D, is trickier because the spinal cord has to be reinserted back into the vertebral canal. That was a quick look at the development of the nervous system. Now what we're going to do is look at the structures that make up this system. And we'll start with the really big stuff. We'll look at the big picture details and then work our way down to the histology and the cytology. So we have two broad divisions to the nervous system. We have the central nervous system, or CNS. This is comprised of the brain and spinal cord. These are derivatives of the neural tube. Then we have the peripheral nervous system, or PNS, and this is comprised of derivatives of the neural crest cells. We have cranial nerves that come directly off of the brain. We have spinal nerves that come off of the spinal cord, and then we have something known as the enteric plexus, which is almost like a digestive system brain, you could say. The digestive system can regulate itself using this network of nerves and ganglia. Like any system, the nervous system receives inputs and it generates outputs. The information is integrated within the central nervous system. So imagine a simple scenario. Let's say you're walking across the road and you notice that a car is coming towards you and it's not going to stop. You need to get out of the way. So we have sensory inputs from the eyes going to the central nervous system, to the brain, and within the brain that information is being interpreted. That's the integration part. Now that's going to result in a motor output, and the motor output is going to be movement in this case. So it's going to affect the proper sequence of muscles needed for you to get off of the road and get somewhere safe. So we have sensory inputs traveling through the peripheral nervous system to the central nervous system, and then we have motor outputs leaving the central nervous system and going out to your muscles in this case. But a motor output could also have an effect on a gland. We have an input, which is sensory in nature, that's going to travel through the peripheral nervous system to the central nervous system. We have integration in the CNS, and that's going to result in an output. A motor signal is going to travel out through the peripheral nervous system out to an effector of some sort. An effector is something that brings about an effect, a change. Could be muscle, could be a gland. Now it's important to note that you are not necessarily consciously aware of the integration that's occurring within the central nervous system. Sometimes you are, sometimes you're not. A reflex, for instance. A reflex reaction, you touch something hot, you withdraw your hand, that doesn't even involve your brain. It only involves the spinal cord, as we'll see later on. Now, in terms of function, we can divide the nervous system into three categories. We have the somatic nervous system. The somatic nervous system is something you're consciously aware of. So we're dealing with sensations that you can perceive, smell, taste, vision, pain, and you're responding to that in a voluntary way. So we have our input that tells you to do something, that tells you to respond to a dangerous situation, for instance. Maybe you're cold, it tells you to put on a sweater. So we have this sensory input, we have a somatic motor response, and this is voluntary, and it's going to affect skeletal muscle, which is voluntary muscle. Next, we have the autonomic nervous system. This is something you don't have conscious control over. So we're talking about fight or flight responses, or what are also known as rest and digest responses. Those are the homeostatic responses, responses that maintain homeostasis in the body. And we'll come back and talk about this in more detail. This is a topic on its own. But note that we can have, for instance, a change in blood pressure. You have receptors within your arteries that can sense a change in blood pressure. You're not consciously aware of this, but if your blood pressure changes, then your body might change your heart rate, or it might change the volume of blood that is being pumped with each beat. Next, we have the enteric nervous system. Enteric means having to do with the digestive tract. I mentioned this before, but you have your own little brain in your gut. It's not a terribly complex brain, but there's a web, there's a network of nerves that connect parts of the digestive system together, and your digestive system can self-regulate using this system. It's interconnected with the other systems as well, but it can work on its own. The spinal cord develops from the neural tube, as does the brain. The brain is essentially just a cranial swelling of the neural tube. The spinal cord is engaged in integration, sometimes independent of the brain. So reflexes can involve just a small segment of the spinal cord without having anything to do with the brain whatsoever. But we also have information that's passing up and down the spinal column. We have impulses that are coming from the brain down to lower regions of the body and vice versa. So the spinal cord serves as kind of an information highway as well. The spinal cord is protected by layers of connective tissue known as the meninges and also by the vertebrae. Cerebrospinal fluid is going to bathe the tissues of the spinal cord and also flow through a central canal that's connected to the cavities within the brain itself. You might be surprised to learn that the spinal cord doesn't extend all the way down to the pelvis. Instead, it ends at your first or second lumbar vertebrae. We have this cone-shaped structure there called the conus medullaris. And then branching off of that, we have these individual spinal nerves that are going to go down to the sacrum and exit through these openings, foramen, within the sacrum. In children, the spinal cord ends at about L3. But of course, children grow and the spinal cord is going to be pulled upwards. As that happens, these spinal nerves are going to extend. We are essentially segmented animals, although that segmentation is entirely internal. We see signs of segmentation in our vertebral column and in our nervous system. And what that means is we just see a pattern of spinal nerves and vertebrae repeated over and over and over again. Remember that each vertebra has a superior intervertebral notch and an inferior intervertebral notch. And when they come together, they form an intervertebral foramen. And that's what you're seeing right here. So that foramen is, of course, a hole that allows a spinal nerve to pass through. The spinal nerves are named after the vertebrae that they're associated with. So we have the cervical spinal nerves, C1 through C8. We have the thoracic nerves, T1 through T12. We have the lumbar nerves, L1 through L5, and we have the sacral nerves, S1 through S5. Now you might be wondering why there are 8 cervical spinal nerves, because there's only 7 cervical vertebrae. Well, the reason for that is that we start with C1 between the occipital bone of the skull and the first cervical vertebra, which of course is the atlas. So because we're starting up above the first cervical vertebra, we end up with 8 cervical spinal nerves. Here you can see a single cervical vertebra removed from a human cadaver, and the spinal cord is still in place. You can see that there's several layers, several protective connective layers around the spinal cord. You can also see the spinal nerves coming off and coming through that vertebral foramen. Down here, we can see the vertebral arteries here and here. Remember that the cervical vertebrae have these holes in the transverse processes. These are the transverse foramen, and they contain this vertebral artery that leads up into the brain and nourishes the brain. Here you're seeing a diagram of the same thing, and I just want to draw your attention to the fact that we have a whole lot of protection. We have these three connective layers that make up something known as the spinal meninges. They protect the spinal cord and they protect the roots of the spinal nerves. Outside of that, we have this epidural space that contains fat that provides further cushioning. And of course, outside of that, we have bone that provides this rigid protection. Let's take a closer look at these meninges. So the meninges are connective tissue and they're coverings, protective coverings around the spinal cord and the brain. They're continuous between the spinal cord and brain. The outermost layer, so the most superficial layer, is the dura mater. Below that, we have the arachnoid mater. It's called that because it's web-like. Arachnoid means spider-like. And then the deepest layer is known as the pia mater. This is tissue that's in direct contact with the spinal cord or the brain. Now I should point out that there should only be one T in mater, and it should be pronounced mater and not matter. It's a common mistake. Mater is Latin for mother. These are mothering tissues. They're nurturing tissues that help maintain the nervous tissue. We also have ligaments. These denticulate ligaments are thickenings of the pia mater that extend to the dura mater. And what they do is they help suspend the spinal cord, kind of float it within the cerebrospinal fluid. So there's a lot of protection here. We have the bony vertebrae, and then we have this epidural space immediately below the bone that's filled with fat. We have the dura mater, which is the toughest of the meninges. It's dense, irregular connective tissue. We have the subdural space, so the space just below that, that's filled with interstitial fluid. We have this arachnoid web of collagen fibers. Then we have a subarachnoid space that's filled with cerebrospinal fluid. And then finally we have the pia mater, which is a very thin layer, contains blood vessels that are going to provide oxygen and nourishment to the nervous tissue, which remember is a very hungry tissue. But also the pia mater will produce these denticulate ligaments that will attach across the void to the dura mater and help suspend the spinal cord within this fluid-filled space. Each spinal nerve has two roots. It has a dorsal root and an anterior root, and these are connections to the spinal cord. The spinal nerve is part of the peripheral nervous system, as is the anterior and dorsal root. So the roots are the connection between the peripheral nervous system and the central nervous system. Note that the meninges extend out onto the roots and help protect them, but they don't extend beyond that. They don't extend onto the spinal nerve itself. Dorsal roots have ganglia, whereas the anterior roots do not. So what's a ganglion? Well, a ganglion is a swelling in a nerve, and this swelling contains the cell bodies of neurons. So neurons, of course, are cells that conduct electrical signals, and these cells can be very, very long. They can have very long extensions. But the cell body, of course, is the part that contains the nucleus. Ganglions contain the cell body. So in blue, you can see the cells that are found within the dorsal root ganglion. We have inputs. We have information coming in from sensory neurons that have their bodies situated within the ganglion. That information is going to be passed to another cell, a motor cell, that's found within the gray matter of the spinal cord. So in the interior of the spinal cord, we have this region that is a different color. It's gray in color, and it contains cell bodies of motor neurons. We're going to have a synapse there. and that means there's going to be information passed from one cell to another, and that motor neuron will send off a signal that will pass through the anterior root. Now, don't worry about this too much just yet. We're going to get to this in more detail. But the dorsal or posterior root contains incoming sensory fibers, sensory neurons, and the cell bodies are contained within that swelling, within the ganglion. Whereas the ventral root is going to be carrying outgoing messages. It's going to be transferring motor information. Here you're seeing an actual section through a spinal cord. And there's a few things I want to point out. First of all, note that we have two deep grooves. We have what's called the anterior median fissure. And I'll just outline that. That would be about here. A fissure is a very deep groove. And of course it's in the middle. That's the median part. And it's towards the anterior. It's towards the front of the body. At the back, we have what's called the median sulcus. And that would be here. A sulcus is a groove that's not quite as wide. It's not quite as pronounced as a fissure. But this sulcus and this fissure effectively divide the spinal cord into two parts. Now, the spinal cord contains white matter and also gray matter. Now, note how I'm using those terms. Again, don't get matter and mater mixed up. The gray matter is going to contain cell bodies. It's going to contain neuron cell bodies. It's also going to contain dendrites and unmyelinated axons, axons that aren't covered in a thick layer of lipid. This will make more sense when we come back and look at the structure of neuron cells. The gray matter is formed into horns. We have what's called the posterior horn here. And then we also have the anterior horn down here. If we were looking at a section through the thoracic region, we would also have a lateral horn. Surrounding that, we have the white matter, which is described as being white columns. Basically, the white matter consists of axons that are running vertically up and down the length of the spinal cord. They're conducting information great distances. We have a gray commissure as well, which is across here. That's this region here, and that's going to conduct information from the gray matter on one side of the spinal cord to the gray matter on the other side of the spinal cord. Finally, we have a central canal in the center here. Remember that the spinal cord starts out as a tube. It never stops being a tube. That lumen gets quite small, but there still is a canal that runs the length of the spinal cord, and it connects to the fourth ventricle of the brain, which is one of the cavities within the brain, because of course the brain starts out as a simple swelling of that neural tube. To summarize, the anterior median fissure and the posterior median sulcus divide the spinal cord into two halves. The two halves are connected through the gray commissure. The gray matter within the interior of the spinal cord is going to contain the cell bodies of neurons, as well as unmyelinated axons, dendrites, and also something known as neuroglia, something we haven't talked about yet. Neuroglia are support cells. They're cells that help maintain the health and well-being of neurons. But this gray matter is going to contain motor neurons. The white matter that surrounds the gray matter consists of bundles of myelinated axons. These are axons of both motor and sensory neurons that are conducting information vertically up and down the spinal cord. Here you're seeing a stained histological section of the spinal cord, and this is through the thoracic region. So you can actually see that there is a third horn It's not terribly obvious, but it's this little lobe here, which is the lateral gray horn. Hopefully you've had a chance to see something like this in the lab. So again, we have a gray commissure. We have an anterior portion, which is anterior to the central canal. We have a posterior portion, which is posterior to the central canal. And note that we also have a white commissure, which is anterior to that gray commissure. And I've just brought up this image again just to point out where that is. So that would be in this area here. And that allows for lateral transfer of information by myelinated axons across the spinal cord. The white matter is divided into columns, as I've mentioned, and the columns are going to contain distinct bundles of axons. These bundles are known as tracts, and they contain axons that will be carrying similar information or heading to the same destination. A nerve plexus is a network of spinal nerves. Spinal nerves come off of the spinal cord and then they fuse together to form major nerves that go out into the limbs, for instance. We have 5 plexuses that originate from the central nervous system. There are 2 plexuses that are especially large, and they come from enlargements of the spinal cord. We have the cervical enlargement that gives rise to the brachial plexus, and then we also have the lumbar enlargement that gives rise to the lumbar plexus. So these are areas where we have a slightly wider section of the spinal cord, and we have a major plexus that comes off and gives rise to major nerves. Here we've got an example of a plexus. We've got the brachial plexus, and the brachial plexus is formed by several spinal nerves. We've got spinal nerves C4 through C8 and also T1 coming together and forming these major nerves that will extend down into the arm. Alrighty, we've looked at development. We've looked at gross anatomy, and now we're going to look at the histology and cytology of nervous tissues and cells. Nervous tissue is made up of nerve cells, or neurons. These cells consist of a cell body, and that's where the nucleus is, and also where the major organelles are. We have projections known as dendrites, and they're going to carry stimuli towards the cell body. And then we have a very long projection that is known as the axon. And that's going to carry a signal to another cell. That other cell might be another neuron, or it might be an effector of some sort. The cells don't connect to each other directly. Instead, there's a gap between them known as a synapse. And we need to have signaling molecules known as neurotransmitters that will cross that synapse and trigger the next cell. We'll talk more about the significance of these structures when we talk about the physiology of the cells. But for now, realize that we have electrical signals, action potentials, being collected by dendrites. These signals are going to travel down to the cell body. If this signal reaches a certain threshold within the trigger zone, this region here, then we're going to fire off an electrical signal, an action potential that will travel down this axon. And it will travel all the way to the end of the cell, and this axon may be very, very long. In fact, in some cases it can be over 1 meter in length. And that signal will travel down all of these little projections here down to the synaptic end bulbs. And that's where this cell is going to synapse with other cells and perhaps excite those other cells, or maybe even inhibit those other cells. The synapse, or synaptic cleft, is a physical space. In the synaptic end bulbs, at the end of an axon, we have vesicles that contain neurotransmitters. If an action potential travels all the way down to this end bulb, These vesicles will fuse with the cell membrane and dump the neurotransmitter into that synaptic cleft. And then those molecules are going to bind to receptors on the next cell in the sequence. Again, it could be a neuron or it could be an effector of some sort. If the neurotransmitter binds to receptor sites, it causes an inflow of ions that cause a change in the electrical potential of the next cell. And that continues this sequence of signaling. These microscopic axons can be incredibly long. So we can have a situation where we have a nerve cell that's in the brain, and that's where the body of the nerve cell is, but the axon extends down the entire length of the spinal cord. It could be over 1.5 meters in length. Materials have to get from the cell body all the way down to the synaptic terminals at the end of the axon. And that's because the materials are being made predominantly within the cell body. That's where we're going to find the Golgi apparatus and the rough ER. So what happens is we have this network of microtubules that run the whole length of the axon. They serve as support structures, but they also serve as railroads. So we have motor proteins that will latch onto vesicles filled with materials, neurotransmitters for instance, and those motor proteins will also attach to the microtubules, and they'll transport this material along that railway network all the way to the end of the cell. Mitochondria are also transported in the same way, and these cells have a lot of mitochondria because they have very, very high ATP requirements. Broadly speaking, we have 3 types of neurons. We have sensory neurons, which are also known as afferent neurons. Afferent with an A. They're going to carry signals through the peripheral nervous system to the central nervous system. And these signals are from sensory structures. Note that the cell body lies outside of the axon. We'll come back to that in a moment. These cells are going to synapse with interneurons. Interneurons, or association neurons, are part of the integration that occurs within the central nervous system. These interneurons are going to synapse with motor or efferent neurons. They're going to carry signals away from the central nervous system through the peripheral nervous system to an effector of some sort. We can also classify neurons based on their structure. First of all, we have multipolar neurons. These have a single axon that leads away from the cell body, but they have multiple dendrites that conduct signals to the cell body. So you can see in this example here, there's several ways that a signal might arrive at the cell body. And if we get sufficient signals coming in, that might cause the triggering of an action potential that will lead down the axon. In a bipolar neuron, again, we have a single axon coming off of the cell body, but we only have one dendrite coming in. There's only one input. Now note that that dendrite has several branches that lead into it, but there's only one dendritic connection to the cell body. Next, we have unipolar neurons, and these are the sensory neurons. The defining feature here is that the dendrite is going to connect directly to the axon, and the cell body lies to the side of the axon. So the signal is not going directly through the cell body. Here are some examples of unipolar neurons, and these are all structures that we talked about very briefly when we talked about the skin. We have Mesner corpuscles, which are responsible for touch. We have Merkel cells. Remember, those are cells that are embedded within the stratum germinativum. These are the most superficial of the touch receptors in your skin. They're responsible for sensation of very light touch. We have Pacinian corpuscles, which are very deep in the skin. They're in the dermis, and they're responsible for deep pressure and vibration. And then we also have receptors known as nociceptors, which are responsible for pain. Finally, we have some extraordinarily large neurons that occur within the brain. pyramidal cells, and Purkinje cells. Purkinje cells particularly are quite fascinating. They have this massive branching network of dendrites that allows them to synapse with hundreds of thousands of cells. These cell types are responsible for cognition. They're responsible for thought. I should point out that the Purkinje cells that we have in the brain are different from the Purkinje cells that are in your heart. Those are actually muscle cells that can conduct electrical signals. You can pretend that you haven't heard of them just yet. That's something we'll get to next term. Here are a couple more images of Purkinje cells, and these cells have been treated with a fluorescent dye to make them especially pretty. But you can see that they are very, very densely branching structures, and these structures are known as dendritic arbors. Dendrite means tree, arbor means tree. They're really, really tree-like. And here's a nice little overview of those different cell types. Bipolar cells tend to be interneurons. Unipolar cells are almost exclusively sensory neurons. Multipolar cells, most commonly motor neurons. And pyramidal cells and Purkinje cells, great big cells that can make multiple connections with lots of other cells to bring about some really extraordinary circuitry that's required for cognition. Nerve cells are very needy cells. They're very, very specialized. They have high requirements when it comes to energy and nutrition. They really can't take care of themselves. And that's where these support cells come in. Neuroglia are support cells. They're smaller than neurons. They're more abundant than neurons, so they actually make up a larger proportion of the nervous tissue. They're not excitable, so they can't conduct action potentials, and they're also not terminally differentiated. Now, what I mean by that is that nerve cells can't do anything else. They become so specialized that they can't engage in other activities, whereas neuroglial cells, in some cases, can adapt to different roles. So let's talk about these support cells, and we'll start with astrocytes. Astro means star. Think of asteroid or asterisk. These are star-shaped cells. They have these projections that come out and can connect directly to neurons. They serve a support function. First of all, they physically support neurons. Nerve tissue is very soft. They can connect adjacent neurons to each other and hold them in place. They also form part of the blood-brain barrier, which we'll talk about more when we talk about the brain. But essentially, they can filter out some of the toxins that might get into contact with these extra-sensitive neurons. They can maintain the proper nutrient and ion concentrations around nerve cells so that they can function as best they can. Something I find really interesting is there's been a number of studies that have looked at the brains of individuals that are especially smart. Einstein, for instance. So there's been post-mortem dissections of brain tissue, and what they found is that these people that had exceptional brain capabilities weren't necessarily sporting more neurons. Instead, they had more astrocytes and more neuroglial cells. They had more of the cells that help maintain healthy neurons. Next, we have oligodendrocytes. These are cells that form multiple phospholipid layers around the axons of central nervous cells. What this does is it forms an insulating layer, kind of, but not entirely, like the insulation that we have around a copper wire. As we'll see, Schwann cells perform a very similar function in the peripheral nervous system. Microglial cells are phagocytic cells that will wander around within neural tissue and gobble up any stray debris. Finally, we have the ependymal cells. These are epithelial cells that line the interior of the ventricles of the brain and the central canal of the spinal cord. And they're going to produce a fluid, the cerebrospinal fluid, that contains oxygen and nutrients and ions and everything that the neurons and also the glial cells need to do their job. And this is just a nice overview of the different cell types we've talked about. We've talked about support cells of the central nervous system, but now let's talk a little bit about the support cells of the peripheral nervous system. First of all, we have Schwann cells. What Schwann cells do is they wrap around the axons of neurons, and they produce phospholipid membrane. And they wrap around several layers of this phospholipid membrane that acts as an insulator or myelin sheath for the axon. Again, very similar to the oligodendrocytes that we talked about before, but these are within the PNS rather than the ANS. Here you're seeing a Schwann cell in action. So you can see it's attached to the side of an axon, and it's wrapping around the axon and wrapping around multiple times. It's creating something that's kind of similar to a cinnamon roll, I guess, for lack of a better analogy. We have multiple layers of phospholipid membrane, and that's going to serve as an insulator. And we'll come back to this in more detail and how this works, how this helps, when we talk about the physiology of nerve cells. Here we can see this in a false color image under the electron microscope. We've got a Schwann cell that has produced many, many, many layers of phospholipid, and that would be the myelin sheath. We also have satellite cells within the PNS. They perform a function that's similar to the Schwann cell, but instead of wrapping around the axon, they wrap around the cell body. So what you're seeing here are satellite cells that have wrapped around the cell body of a sensory neuron. And that allows them to control what materials get in and out of that cell. Just as a reminder, in the central nervous system, we see gray matter and we see white matter. White matter consists of myelinated axons. These are axons that have been coated in multiple layers of phospholipid by Schwann cells. And then the gray matter consists of the cell bodies, but also the neuroglia as well. And here's our terminology. In the next topic, we'll delve into the function and physiology of nerve cells. You'll love it.